Okay, let's dive right into this. Welcome to today's explainer, where we're putting a magnifying glass over one of the absolute classic showdowns in O-level biology. I actually like to call it the water battle royale. On one side of the ring, you've got the sturdy, heavily structured plant cell, and on the other, the flexible, kind of squishy animal cell. We're going to drop both of these into completely different watery environments and watch exactly what happens at a microscopic level.
So here is the core mystery we're solving today. Why doesn't a plant cell burst when you put it in water, but an animal cell absolutely does? If you're studying for your biology exams right now, nailing down this specific structural mystery is absolutely crucial. It's literally the key to unlocking major marks on questions about cell structure and fluid dynamics. We aren't just going to memorize textbook facts today, no way. We're going to visually track the exact physical forces at play so you never ever forget the mechanics behind the answer.
To really get our heads around this, let's start with a macro analogy. Think of a basketball. I want you to visually map this rigid, tough leather sphere to our plant cell. Got it? Now, think of a highly flexible water balloon and map that directly to our animal cell. Both of these objects hold water, right? But notice the structural boundary markers. If you hook a hose up to both and pump them full of water, they handle that intense internal pressure entirely differently. The water balloon just expands until its thin barrier completely snaps. But the basketball, it just gets incredibly firm. Keep these two physical objects in the back of your mind as we shrink down to the cellular level.
All right, Section 1, the rules of the game, osmosis and water potential.
Rule number 1 is osmosis. Simply put, this is the diffusion of water molecules across a partially permeable membrane. Now watch the directional arrows tracking the flow of water here. It always moves from an area of higher water potential to an area of lower water potential. It is a strictly directional flow.
And that brings us to rule number 2, water potential. You can think of this as a measurement of the literal freedom that water molecules have to move around. Pure water has the highest possible water potential. But watch the visual scale indicators as we drop little solute molecules into the mix. See how the potential drops? Adding solutes literally decreases the water's freedom to move.
Okay, let's briefly pause our flowchart here because we need to clear up a couple of really common misconceptions that trip students up all the time. First, a lot of people think all cells burst in water. As we'll see in a second, that is simply not true. Second, we have to use precise vocabulary. Water potential and concentration are related, sure, but they are completely distinct concepts. When we're talking about water moving, you've got to train your brain to think in terms of water potential gradients, not just general concentration.
Moving on to Section 2, Arena 1, the hypotonic environment, or distilled water.
All right, here we go. We're dropping an animal cell, let's say a red blood cell, into pure distilled water. That means we have the highest possible water potential on the outside. Step 1, high water potential outside the cell. Step 2, because of that gradient, follow the inward directional arrows. Water rapidly enters the cell via osmosis. Step 3, look at the dimension lines expanding. The cell volume swells up incredibly fast, just like our water balloon. And step 4, the pressure is just too great. Pop! The membrane bursts. This total destruction is called hemolysis. Our animal cell is officially out of the game.
Now let's contrast that by dropping a plant cell into that exact same distilled water. Step 1, high water potential outside. Step 2, water enters via osmosis. Step 3, the cell volume starts to expand. But watch Step 4 really carefully. The plant has a sturdy, rigid cell wall. As the internal volume expands, notice the visual boundary markers. That thick wall is exerting an equal and opposite force, physically pushing back inward. The cell doesn't burst, it just becomes incredibly firm, a state we call turgidity.
And this brilliantly illustrates the macro consequence of these microscopic rules side by side. The animal cell undergoes hemolysis and gets destroyed because it completely lacks a rigid boundary. Meanwhile, the plant cell achieves turgidity, maintaining its structural integrity perfectly, all thanks to that rigid wall pushing back against the osmotic pressure.
Let's change things up. Section 3, Arena 2, the hypertonic environment, our concentrated salt solution.
Let's plunge our animal cell into a concentrated salt solution. Step 1, because of the salt, the water potential outside the cell is now lower than the inside. Step 2, look at the arrows. The flow has completely reversed. Water is now exiting the cell via osmosis. Step 3, watch the scale dimensions shrink as the cell's volume decreases dynamically. And Step 4, the whole cell just shrivels up. This physical shrinking process, that's called crenation.
Okay, Section 4, acing your biology exam.
So, we have the visual mechanics locked in, but now let's translate all of that into the exact words examiners are actually looking for.
Here's a super classic O-level exam prompt for you. Explain why a plant cell does not burst in distilled water. You know the visual steps, but how do you write it out to guarantee full marks?
We're going to explicitly map the visual vectors we just learned directly to your exam keywords. When you visualize that high freedom outside the cell, you have to write high water potential. When you picture those water arrows pointing in, write enters via osmosis. For that thick boundary marker pushing back, you absolutely need to write rigid cell wall exerts an equal and opposite force. And finally, for the cell staying intact, use the magic exam keyword becomes turgid. Connecting the visual concept to this specific vocabulary is exactly how you ace this.
Let's do one more rapid-fire test of your active recall for the hypertonic arena. The prompt is, what happens to an animal cell in a concentrated salt solution? And here is your grading checklist. First, state the condition. The water potential outside is lower. Second, state the action. Water exits the cell. And third, the result. The cell crinates or shrivels. If you hit those 3 points, the marks are yours for sure.
So the crucial point I want you to take away is this. When you're dealing with plant cells in an exam, your absolute secret weapon is 2 words: cell wall. That detail alone is usually the difference between partial credit and scoring full marks. Whenever you get stuck, just close your eyes and visualize that macro analogy from the very beginning. Visualize the resilience of that basketball pushing back.
We've covered a ton of ground today, tracking the vectors of osmosis and the vital importance of structural boundaries. But I want to leave you with a final thought to ponder. If a plant cell absolutely relies on that internal water pressure pushing against its rigid wall to stay turgid and upright, what happens structurally to a whole living plant when its cells are placed in a concentrated salt solution? Imagine those outward shrinking forces we saw earlier, but on a massive scale. Visualizing the mechanics of that phenomenon, a process called plasmolysis, is your next great biological mystery to solve. Stay curious, keep visualizing those mechanics, and I'll see you in the next explainer.